Pleasure to introduce Sepide Sadayani. Despite of what you see on her Zoom, she goes by her name with which she publishes. So Sepide got her bachelor's at the University of Osnabrück in Germany, as well as UC San Diego. Then she did a PhD at Max Planck. Followed by a postdoc at UC Berkeley with Mark Esposito, and I had the honor of doing a postdoc at the same lab, so we interacted there for a while, and we even shared a space. We sat next to each other, and that was like, a really good experience for me. I learned a lot from Seppi. She followed that by another short postdoc at Stanford, and then became an assistant professor at University of Illinois Urbana-Champaign since 2016. Sepide has been extremely successful. She has already a R1 grant and another R21 grant as a PI, and she publishes really well. What she works on is this combination of fMRI and EEG and IEG, and I think she's going to tell us about that, as well as network properties of the brain. Really looking forward to your presentation, Seppi. The floor is yours.
Thanks so much, Toby, and thanks so much for inviting me. It's exciting to at least be virtually connecting with you. So as Toby mentioned, I will be talking about integrating fMRI and EEG to learn specifically in this talk about the large-scale functional connectome. And I hope that I will be able to convey the message that really comparing the modalities is going to tell us more than each one alone. I just want to start with thanking those from whom I will be presenting data today, as well as lab members that are currently collecting scalp EEG fMRI data. A lot of the data that you will see in this talk are actually coming through collaboration. So I'm extremely thankful for the generous data sharing here, as well as collaborations on methods. Okay, so I'll be starting with an introduction of what the intrinsic connectome is, and focusing on fMRI because that's the method of choice by far to look at the whole brain connectome. I will then move on to hopefully convince you that electrophysiological measures can be used to study the intrinsic connectome in a meaningful way. And then the second section of the talk will be about the multimodal integration of fMRI and electrophysiology, first looking at the so-called static or time-averaged connectome and focusing on the spatial relationships, and then moving on to time-varying dynamics and how they may or may not co-occur across the modalities. So I'll start here with the notion that the brain is highly active, irrespective of what the person is doing at a given moment. So you're probably all very familiar with this argument by now that the brain is consuming about 20% of the body's energy, irrespective of whether it's actually mentally engaged, resting, or sleeping even. So this is a PET image of glucose consumption, and you can really appreciate the high energy demands compared to even other energetically demanding organs. So the way we typically tend to think about evoked brain responses, or the way we generally have been studying the brain for a long time using neuroimaging and electrophysiological methods as well, is that we provide an external stimulus, and somehow this goes through our senses, and then the brain shows what we call prototypical or maybe veridical evoked response. But if you actually look at the trial-by-trial response, you will find an enormous amount of variability, which is the very reason why, for example, ERPs need to be repeated over many trials. So event-related potentials are really, by definition, trial averaged. And the average really looks like a textbook signal. This is actually intracranial recording from primary auditory cortex. But I just wanted to show you how the individual evoked responses may be very variable from trial to trial of the same sound, and how much background activity there is actually in these baseline fluctuations. So when I talk about intrinsic, what I mean is simply all of the brain's activity that is not directly evoked by an external stimulus. So just for clarifying the definitions, and a lot of what I will show today are looking at connectivity matrices. So the way those are constructed in fMRI is that you typically have a whole brain parcellation of the brain. There's various ways of using or defining these parcellations. And for any given brain pair of brain regions, we look at the co-fluctuation of the bold signal. So between these two regions, you would, for example, see high temporal concordance, which would result in a high Pearson correlation value. And there are regions then that also have very low correlations. So looking at the resting brain, or in any brain state, really, you can retrieve, based on these low and high correlations, these so-called canonical intrinsic connectivity networks, for example, for the motor, auditory, visual, dorsal attention, and so on and so forth. And really, this is a spatial independent component analysis of the resting fMRI activity. You will see that the brain is intrinsically organized in these canonical networks that actually do reflect the same co-activation patterns we typically see during any given task. And if you align these regions of the parcellation along the regions that build these intrinsic networks, you get this whole brain connectivity matrix, typically in fMRI representing Pearson correlations, and you will see along the diagonal these canonical networks organizing. So the whole brain connectome can be thought of as something like a communication network of the brain, which we can study also using graph tools, similar to ways as that we use for social networks, for example, in topological space. Now, an important property of the connectome as observed with fMRI is that it's incredibly stable over mental states. So this has been shown in numerous different studies. Here to the right, you see an average across a large number of subjects, an average connectome over task-free resting state. To the left, you see a subject average connectome and averaged over numerous tasks. And the spatial correlation between these two spatial patterns, these two organizations of connectivity, is extremely high. However, over and above this stable organization, there are subtle changes in the connectome from moment to moment, these so-called time-varying dynamics. And I'm just illustrating here why those actually matter. So this is an example study where we show that those subtle changes in the connectome's pattern from one moment to the next is actually impacting behavior. So this is a cartoon of the hemodynamic response to a given sound, and we have a number of experiments where we spread out our trials in extremely sparse manner. So these are 20 to 40 seconds inter-trial intervals, which give us these windows that are free of the hemodynamic response. So we then built these all by all kind of correlation matrices from individual windows prior to trials. So the correlation matrix might have subtle differences from one occurrence to the next. And if we feed these into a machine learning algorithm, we can actually predict above chance. So this is the distribution of chance accuracy of the prediction. We can predict with above chance accuracy whether or not the subject is going to perform well on the next trial. So in this particular experiment, the stimulus was a threshold level sound. And so because it was so faint, the subject would miss it on half of the trials and hear it on the other half. And whether or not the subject is able to perceive the sound depends on these pre-stimulus states. We have replicated similar findings in other sensory modalities and other types of stimuli. Another aspect of the study is that we can build these topological graphs, and I'm showing those for one subject here, prior to successful detection of the sound and prior to missing the sound. And we found that the organization into these canonical intrinsic connectivity networks is more stable, is more segregated. These networks are better defined prior to hits compared to misses. Also at the group level, you can see that prior to the misses, the spatial organization into the intrinsic networks breaks down to some degree compared to hits and compared to task-free resting state. So this is just to mention that in spite of the stable organization, we really are seeing a behavioral impact, a significance of the intrinsic connectome. Okay, I'm now moving on to electrophysiology. So the most typical traditional view of EEG and fMRI is that there is some level of neural activity aligned between a large enough number of neurons that generates electrical or magnetic signals that can be picked up by electrophysiological measures over the scalp, and that the same neural activity through the hemodynamic coupling generates signals that can be picked up by fMRI. So there's this idea that there is a shared neural basis, and then there's, of course, modality-specific noise, which will result in some level of discordance when we compare these signals directly with each other. In the context of connectivity, one particularly important source of noise for non-invasive measurements is signal leakage. So unfortunately, the underlying electrical and magnetic signals are actually spreading over space in such a way that at sensor level, we're not sure where those signals are actually coming from. And so we have this mathematical inverse problem that we need to go from the sensor space into the source space. And this is perhaps the major reason why people have shied away from using non-invasive electrophysiological measurements in order to look at large-scale connectivity. Another aspect that perhaps causes caution is the very asset of electrophysiological measurements, and that is that they are so rich compared to the fMRI signal. So in particular, you may have brain regions, for example, here, B and A, that couple to each other with respect to the phases of their ongoing activity. So much of ongoing activity is thought of as being oscillatory over and above the sort of 1/f distribution of the background signal. So this oscillatory aspect is typically the part of ongoing activity that is used in EEG to build connectivity. So regions can be coupled with respect to the phases of those oscillations, or alternatively with respect to the amplitude of those oscillations. So for example, these two regions might be coupled in terms of amplitude of a given frequency, but not in terms of phase. But then of course you could also have both, where a region pair can have phase coupling and amplitude coupling at the same time. To make matters more complicated, EEG is actually, or all electrophysiological measurements are actually spanning 4 orders of temporal magnitude, anywhere from infraslow, which is what's typically recorded in the bold signal, and if you allow your amplifiers to actually record that range, all the way to the range of the canonical frequencies up to 100 Hertz and higher. So we can define connectivity in various ways when it comes to electrophysiological signals. In spite of the methodological difficulties of signal leakage, I'm just illustrating here one of the general pipelines that can be used and has successfully been used in order to look at whole brain connectomes using scalp data. So from the raw EEG signal, we will typically want to filter the signal in these canonical oscillation ranges, although there are studies that are looking at the broadband EEG signal as well. We then source localize the signals from the scalp onto a mesh of individual sources in the brain, and then average those source signals across a given brain parcellation, similar to how we would average voxel-wise bold signals across regions of an atlas. Then we can look either at the actual phase of the signal or the power envelope, and estimate phase coupling or amplitude coupling, as I just mentioned, in order to retrieve these whole brain connectivity matrices and then do any other computation as we would regularly do with connectomes retrieved from the fMRI. There are some other steps necessary that unfortunately have to be over-conservative and throw away a lot of the good signal just because we want to be sure that we're not looking at spurious connectivity that is coming from the same, is based on the same signal projecting to a distance location. So I'm happy to discuss this if anybody is interested, but the major point I want to make is when these cautious steps have been taken, there is a mounting amount of evidence from various different labs that we can meaningfully look at the whole brain connectome using EEG and MEG. So this is an MEG study actually at resting state showing in a seat-based manner, so seat-based correlations that are typical in fMRI, that we can retrieve a good number of these intrinsic networks. Similarly, using temporal ICA on resting state MEG, you see this astonishing similarity between the spatial independent components that we are so familiar with in fMRI, this is always the top row, compared to the matching components from MEG. And you see various different networks here, both the primary sensory and motor networks, as well as the higher order networks, such as the default mode network, the left and right executive control, and so on. So just to be sure that we can establish some of the major points we want to make, in the absence of signal leakage problems, I first moved to intracranial studies here, and then I will move back to scalp a little bit later on. So this is an example of how electrocorticography may look like in a patient where of course, these human subjects are undergoing surgery for medical reasons, and typically that will be intractable epilepsy, where patients are not well responding to medication. During the time that they're in the hospital, this takes a week, 2 weeks, depending on the subject. We have a lot of time to collect tasks with these patients. So the dura is opened up, and then electrodes are placed directly on the cortical surface, like here. And of course, we don't get whole brain coverage, so that's one of the major drawbacks of electrocorticography. But I'm just plotting here 3 example subjects from this publicly available data set that we've used for this particular study. And you can see that these grids are actually covering more than one lobe. So very often, at least temporal and frontal lobes, sometimes more parietal areas and so on. But we have coverage of various different functional areas. So for one particular subject, I'm here showing the connectivity matrix using phase locking values. So this is phase-based connectivity in the theta band for all the electrodes that we had available for this particular subject. And the point that I want to make here is that when you look at a task-free resting state versus a very cognitively demanding 2-back working memory task, you see quite a bit of spatial similarity. And for this particular patient, we had a much higher similarity even looking at, for example, the beta band. So now summarizing the similarity across mental states, we really come to a similar viewpoint that has been previously established for fMRI. What I'm showing here to the left are spatial Pearson correlation values. So there are these, let me actually turn on my laser pointer, making sure you can see this. So these spatial correlation values that I'm showing here are plotted in every element of this matrix. And then I'm going through the different mental states. I have task-free resting state. I have pre-stimulus baseline from 2 different tasks. And then I have 3 very different tasks. This is finger tapping, verb generation, and 2-back working memory. The other aspect I'm showing here are the different canonical frequency bands here, side by side. So along the small diagonals, you always see the within frequency similarity across mental states. And then off diagonal, you will see the cross-frequency similarities. So these values are extremely high. This is average across anywhere, depending on the comparison, anywhere between 4 or 5 subjects to 18 subjects. We also assess this, of course, within individual subjects and individualized Nile models. And the spatial similarity is significant in almost every single subject and almost every single comparison. That's what we're showing here in the bottom. So another way to look at the same data is 1 minus the correlation value. So I call this the similarity here. The point that I'm trying to make here is that the similarity of resting state to resting state, so we just split the data in half, is not any stronger than resting state to the tasks, or even any task to another task. So again, this radar plot is just making the point that what we know from fMRI in terms of the stability of the functional connectome holds true in electrophysiological measurements. Another point is the similarity between the various ways that connectivity can be defined in ongoing oscillations. So to the left, I'm showing amplitude coupling, and to the right, I'm showing phase coupling. This is measured as phase locking value in terms of phase lag consistency over trials. This is during task performance, and this is a post-stimulus activation window. These are just 4 example subjects here, and I'm going to show you the spatial correlation between this matrix and that matrix in the following slide. So again, we have a stimulus here in a given task, and then we're looking at the correlation matrix retrieved from the post-stimulus time, either in terms of phase coupling or amplitude coupling. That's what I'm showing here in the black lines. Every single black dot is 1 patient. So this is spatial correlation for different canonical frequency bands. In the post-stimulus period, this is the period that is typically looked at when we're looking at electrophysiology. You see a moderate size, but in every single subject, significant correlation. Here are the null models at the bottom. But what's interesting also is that the correlation between phase coupling and amplitude coupling is already present in the pre-stimulus baseline. So if we build the connectivity matrices now from the baseline here, and then look at the spatial correlation between amplitude and phase coupling, this is shown in the gray color here, it's almost identical to the post-stimulus period. If we first subtract this matrix from that matrix, so we just have the difference matrix of what changed in the connectivity pattern from baseline to task, we get the orange line, which is extremely low spatial similarity. So really all the similarity between the 2 connectivity measures is already present irrespective of the task. So it's not task driven, it's really intrinsic to the brain. So just to summarize what I showed you here in the static connectivity section is that the functional connectome can be meaningfully studied using scalp and intracranial electrophysiology, that electrophysiological connectomes are very stable across frequency bands and across coupling modes, phase coupling and amplitude coupling, and that they are very stable across mental states. What I showed is fMRI and intracranial. Oh, sorry, I didn't actually show the fMRI yet. That's yet to come. Sorry. So the fMRI from the background literature as well as the intracranial data that I just showed. So now I'm actually moving to the multimodal connectomes. I will first cover the static connectome. So this is the time averaged connectome, and then I will move on to temporal dynamics. So as mentioned earlier, we can use a similar atlas or brain parcellation for source space localization of the EEG signal, similar to the atlas that we use for fMRI. So if we use the same atlas, and unfortunately it has to be a rather low resolution one because of the limitations of EEG source localization, but if we use the same atlas for both modalities, we can establish a static or time averaged connectome per subject for the fMRI, and one for EEG for any canonical frequency band. So this is older data by Jonathan Bierzig, and he has shown that for the subject averaged connectome, you find a reasonable moderate size correlation, spatial correlation between the fMRI edgewise connectivity strength and the EEG edgewise connectivity strength. So in this case, I'm showing an imaginary coherence from the beta band, but similar values were observed for other frequency bands. So every dot in this plot is one connection of that connectivity matrix. So we're just plotting the strength of those connections across the modalities. A particularly interesting piece of data that really convinced me to go into looking at connectomes with scalp data. So this is scalp EEG and concurrent fMRI, is that the relationship between EEG connectivity strength and structural connectivity is at least as strong as that observed between fMRI and structural connectivity. At least for the atlas used here, with, of course, all the limitations that come along with such comparisons. But this has been replicated, or this has been previously shown also in other studies using amplitude coupling, again, also with concurrent EEG and fMRI by other labs. So the structural connectome here is estimated by tractography from diffusion-weighted imaging, so also non-invasive. The fact that the correlations are negative is just based on the way the connectivity is defined here based on average path length. But important is here the effect size. So again, this is the subject averaged connection strength. And the reason I'm emphasizing this is in the early days when fMRI resting state connectivity came along and people were very skeptical to what degree it might be driven by vasculature and other sources of noise that affect fMRI. One important piece for the field was this observation that the fMRI-based connectome has this relationship to the underlying brain structure. And that was convincing a lot of people that it is worth looking at the intrinsic connectome using fMRI. So if that argument is taken at face value, then we should feel fairly good about the fact that in spite of source leakage, there is enough information in the whole brain EEG connectome to work with. Another really important observation we made recently is that when you build these correlation values between fMRI and concurrent EEG, you seem to always see a small to moderate effect size around 0.3, no matter what the setting and what the data processing. So we've established this now between 1.5 Tesla all the way up to 7 Tesla, from 64 EEG channels to 256. Anywhere from 10 subjects to 26 subjects in every data set. And no matter what we do, we seem to have across all the EEG frequency bands the same level of spatial correlation. There just seems to be a natural cap that we just can't overcome. And it might be even more visible in this plot here, where we're showing the similarity between the spatial similarity between EEG and fMRI-derived connectomes as a function of the number of subjects going in. And you see that for all frequency bands, anywhere between 7 and 12 subjects will cap the correlation at this medium range. And in fact, above 12 subjects, we don't gain more than 1% of correlation of the effect size, just moving on and adding more data. We observed the same thing by the fact that no more than 5 minutes resting state is actually going to improve this similarity. And also using different parcellations of the brain isn't helping anything. And so I'm showing here data for phase coupling. We saw something very similar for amplitude coupling. There seems to be this natural barrier that we can't overcome that somehow makes me feel that we're not just fighting methodological noise that causes the discrepancies between the modalities, that there might be more to it. Another piece of information that really convinces me of this viewpoint is this study by Denny Bassett. They are looking at intracranial recordings and offline bolt signals from another set of subjects. What they're doing here is actually looking at the entire whole brain connectome by merging whatever edge is available from the given patient across about 100 patients. So they're taking whatever piece of the connectome they can find in any given subject, and they put them together and compare them to a subject average fMRI connectome. They seem to see a similar range of connectivity values, no matter what frequency range they're looking at. So very similar using intracranial data in the absence of source leakage. We also wanted to move on beyond correlations and actually do a data-driven decomposition of bimodal concurrently recorded scalp EEG, scalp fMRI here. So what we've done here is building a static connectome per subject for fMRI and then per subject per frequency band for EEG. We basically just vectorize the fMRI and EEG connectome and glue them together, and then we stack the subjects and frequency bands. So this is ICA as you would typically know it, except that typically ICA is used on the signal time series, but here we're using ICA on the connectivity values. So this is has been coined as con-ICA. It's ICA essentially of these connectivity patterns over subjects. So what we're asking here is, what patterns of the static EEG and fMRI connectome are co-expressed over subject? So we call those hybrid traits. We found replicated across two different data sets at 1.5 and 3 Tesla and various different recording settings. We find one very stable hybrid connectome trait, which has a similar, relatively similar spatial pattern between fMRI and EEG. So we show using modularity, for example, and other approaches that these patterns relatively well conformed with those spatial organizations that are stronger within the canonical networks, as well as in contrast to across. So there is a significant spatial similarity between these, and they seem to recap essentially the intrinsic organization of the brain networks. Here I'm just showing the subject-specific and frequency-specific weights of these patterns. They're pretty broadly distributed across all frequency bands. But what I actually want to emphasize here is a second trait that we found. So we found two very stable traits that replicated across data sets. And this second trait spatially has nothing to do between EEG and fMRI, even though by definition it is co-expressed. So this represents a hybrid bimodal trait of the connectome, but is spatially extremely discordant between the two methodologies. Another piece of information that seems to suggest that there is something substantially different happening between the two modalities. So let me close with the last section here by moving to time-varying dynamics. Dobie, is it okay if I talk to the full hour?
Yeah, you're good.
Thanks so much. So I'm sure you have heard about connectome dynamics in other settings. It is becoming more and more of interest to the neuroimaging community to look at how the changes of the connectome can be characterized. And the reason that matters is the study I showed early in the introduction, that kind of dynamics seem to actually impact behavior. So we typically look at the connectome the way I showed it up to now, which is building an average connectome by putting in all the data points from the entire recording. However, one could build these time-varying connectomes by essentially having shorter windows from which the connectivity is built. And at certain time points, you might find connectomes that are pretty similar to the static connectome, but at other times, there might be quite a bit of deviations from what the so-called average connectome will look like. And this has been hypothesized to actually represent so-called cognitive architectures or cognitive modes that each might be supportive of a different kind of processing setting, essentially, that might be good for particular tasks and bad for other tasks, and that the brain is exploring these patterns in an iterative manner. So this has been, again, studied almost exclusively in fMRI, but of course, EEG is the method of choice when we're interested at anything temporal. It has much better temporal resolution. So what we have done is applying these typical sliding window connectome approaches for concurrent scalp EEG and fMRI. So in this case, we have a 60-second window because we're using Pearson correlations, and we need, unfortunately, enough time points to build a stable connectome at a given point, and we are stepping at the resolution of our fMRI recording. We do the same thing for EEG, so we build imaginary coherence connectomes in source space, and then average the coherence value for a 1-minute window and then step over the same time. We build mutual information for every edge of this connectome and compare that to an appropriate null model. So we're asking for every edge of the connectome whether the change in connectivity over time is accompanied by change in connectivity in fMRI. And I'm showing here subject group T values. Essentially, almost 100% of the connections of the connectome are surpassing the null model in terms of showing codynamics between EEG connectivity fluctuations and fMRI connectivity fluctuations in task-free resting state, giving enough data. So we had a replication data set that had fewer subjects and shorter recordings, and we see about 1/3 of the connections passing the null model. So this is passing at Bonferroni corrected, by the way, for over 2,000 connections. So it's incredibly strong relationships if you have enough data. But I have to emphasize this is evaluated for every single edge separately. So we're not looking here at the connectome distribution or spatial pattern overall. But the major point here is that I'm just here plotting the top 200 edges for every frequency band and its relationship to fMRI. So you see that it's not the case that this top 200 are restricted to a certain section of the connectome. It looks like every frequency band really has connectivity distributed across the entire connectome here, shown as a circle graph. So I'm just essentially plotting all of those parcels around the circle here. There are differences. So if you look at how the edges are distributed over the different region pairs, there are definitely differences. But the major point here is that the spatial patterns are extremely distributed over the entire brain. We wanted to improve on this study in 2 ways. First of all, as I mentioned, our previous study was just looking at every edge and asking whether changes in the connectivity strength of that particular region pair in the brain are related between EEG and fMRI. We now wanted to look at this pattern of the connectome with this notion that there are these discrete, quasi-discrete connectome states that the brain is cycling through, which means that the actual spatial pattern matters. So in the next study, we wanted to move to spatial correlations in every given window of time. The other thing we wanted to improve on is moving away from Pearson correlations, is having an instantaneous connectivity measure for fMRI, which afforded us much shorter windows. This next study is actually, I'm going to first show intracranial concurrent EEG and fMRI. And the reason I'm emphasizing this so much is that this data set is absolutely unique in the world, and having concurrent measurements from the human brain intracranially with fMRI. And I'm really fortunate and thankful to the collaborators from UC London that have generously shared this data with us. So again, what we've done is, okay, interesting. Let me let me try to circle back and see if I can get this started. So what I'm just illustrating here is visually the calculation that we made. So we generate, again, in every single window, we generate a connectome for every patient. And do that for bold and concurrent amplitude coupling and phase coupling. And then at every window, we assess the two-dimensional spatial correlation between fMRI and EEG. So here's the method illustrated again. We have single window fMRI connectomes and single window EEG connectomes. And then we just plot the spatial Pearson correlations over time. What is grayed out here is the level dictated by an appropriate null model. So only these dark black sections are actually surpassing our null model for a spatial similarity on a single frame, essentially, between EEG and fMRI. I'm going to quantify this in a minute so you can have a better look at it. But first, I wanted to say that if we take a different approach, sorry, here the time series here is the same two-dimensional Pearson correlation between fMRI and phase coupling, measured as phase locking value. And these gray areas are the time points that surpassed that null model level. In the bottom, I'm showing an alternative approach using k-means clustering of discrete states. This is one of the standard approaches, either hidden Markov models or k-means clustering of individual windows of connectivity, so that you can have a data-driven way of defining these quote-unquote discrete states of the connectome. So color coding just shows here connectome pairs between EEG and fMRI that co-occur. For example, in this color, you would have a connectome pair that actually is aligning with the times of significant cross-modal spatial relationship and may co-occur between the two modalities at later time points as well. So essentially, this plot is showing that the time points in which there is an above chance spatial similarity between EEG and fMRI co-aligns with a separate method that actually looks at a data-driven way of finding discrete states within the connectome dynamics. So as promised here, the quantification of the number of time windows that actually surpass the null model, and it is shockingly low. We have tried this with different window widths. We've tried this using amplitude coupling and phase coupling. And most importantly, we have replicated this using scalp whole brain connectomes of the healthy brain using EEG concurrent fMRI. So we see the exact same picture, not just within small patches of the cortex, but using the whole brain connectome and, of course, using the healthy cortex as opposed to the patients. So there seems to be something true about the shockingly low occasions in which the spatial pattern of the connectome meets between EEG and fMRI. And I'll close with that by summarizing that there is a cross-modal similarity of the static connectome organization between fMRI and EEG. The moderate effect sizes around 0.3 that I was showing were if you actually average the connectome within modality first across subjects. So you have a subject average prototypical connectome in fMRI and a prototypical connectome in EEG. Those share a moderate size relationship. If you look at this with individual subjects, you actually find anything between 0.1 and 0.2 correlations, both for scalp EEG concurrent fMRI as well as intracranial EEG and fMRI, as well as offline MEG and fMRI from other labs. They have confirmed this small effect size that we just don't seem to be able to surpass for some reason. The cross-modal association of connection-wise, infra-slow dynamics, so in those 60-second very slow windows at a single connection evaluation shows a significant relationship between the modalities in almost every edge of the brain and in every frequency band. However, the whole brain connectome states, the patterns, seem to meet across the modalities only on rare occasions. And the same spatial patterns seem to be explored by fMRI in EEG. So the spatial patterns that they show in a dynamic fashion are actually spatially similar. They just don't co-occur in time. One reason for that is that the EEG is cycling through those states much, much more rapidly than fMRI, and I have not shown that data here, where I'm happy to discuss it or show some figures. So that's really exciting work that is ongoing right now to show that there is a spatial concordance, it just isn't temporally concordant. And so I'm left with this notion that as much as I want to look for commonalities, the data is speaking a different language to me. The data is suggesting to me the more I look, the more robust is the divergence between fMRI and EEG. And I want to suggest that that may actually have some neural reasons. It's not just the noise in EEG and fMRI that's contributing to the cross-modal divergence, but there might be an alternative view that there is some level of shared neural activity that comes from the same neural sources and is reflected in both modalities. However, there is also neural activity that might be weighted much more strongly in EEG, and conversely, other type of neural activity that is weighted more strongly in fMRI, and they don't really overlap much in our measurement. So I want to close with this direct quote from one of the papers of Rita Hari. She's suggesting MEG and AEG weights strongly the fastest conducting pathways, whereas fMRI probably receives its main contribution from neural ensembles that are connected via slow and thin fibers, thereby apparently reflecting functionally different brain activations. So I'm tending to think about this as having these really thick myelinated fibers, like the highways here. And then we have in the brain also these thinner, slower, less myelinated fibers. And they're intertwined, and sometimes they might even run in parallel, such as frontage roads that are running for large sections and parallel to the highways. But you might say, well, why would I care unless I'm doing this kind of research that there is some kind of stuff that can be picked up by fMRI and other stuff by EEG? So why would I care unless I have a methodological interest in this? Well, it may suggest that the brain is fundamentally a multi-timescale system. And the reason that that's the case is because different cognitive processes are actually unfolding at different speeds, out of necessity, dictated by nature. And that argument is beautifully made by Rita in this talk that she gave in the Talarak lecture in Rome at the last in-person HBM that we had. So I shrunk that URL for you. I highly recommend this. She's not focusing on connectivity, but much more generally activity in the context of cognition and how cognition can be slow or fast, and that we need both, and how those speeds are reflected differentially, potentially in different modalities. And I would like to leave you with that conclusion, and thank you again for being here today and for having me.
That was a wonderful talk. So if anybody has questions, please put them in the chat, or if you want to unmute yourself and ask, feel free. Let me start with a quick question on my end, and if anybody wants to jump in. Actually, we already have a question, so let me go with that. So Seppi, I don't know if you can click on the chat and actually see it yourself as well. So the question is, as both signals is indirect measurement of neural activities, would one propose EEG signals would represent earlier fluctuations of brain than of fMRI?
Yes, so that's an important point. When we directly try to compare the concurrently recorded measurements of EEG and fMRI, we shift the EEG by 5 to 6 seconds, which is about the time that neural activity takes to peak in electrophysiology. Depending on the studies, one might argue that one should also HRF convolve the EEG. We have tried different ways convolving and not convolving depending on the research question. But we definitely need to do that temporal shift. That is absolutely true. And that doesn't fix the dissimilarities if that's the point you're making. Again, I'm emphasizing the dissimilarities today just because I think that they have not been really discussed in a conceptual way so far. But I have shown you hopefully convincing evidence that there is a whole lot of similarity across the modalities to work with. And we're really excited about those concordances over space and time as well. But in terms of time, we definitely need to account for that delay, yes.
Great. I was wondering, can you do modeling on this stuff? So it seems like it's going to be difficult to make a very convincing argument about this, are EEG and fMRI actually predicting different things? Is it the white matter connections? Is it something else? I think all of these things, at least in principle, should be implementable in models, and you can then simulate the model and see what you get.
Yes.
I think it's difficult.
Yes. So there is one set of models that might work in the direction that you're thinking, and those are generative models where typically the structural connectome is taken as a ground truth or driving kind of organization. And then there is a Kuramoto model or some other way of coupling oscillators, some model of simply coupling, feeding signals into the nodes that are connected via that structural connectome, and then see what the predicted generated electrophysiological connectome would look like based on that backbone. And also what the functional, the fMRI-based connectome through the hemodynamic lens would look like. In terms of effect sizes, I would have to go back to those. They definitely show similarities. So that approach can definitely, if you generate an electrophysiological connectome, assuming that what fMRI is picturing is actually electrophysiology passed through HRF, if you do that, you indeed retrieve a pseudo fMRI connectome that's fairly similar to the observed connectome. But in terms of the similarity between that fMRI and the electrophysiological connectome and the dissimilarities, I would have to go back to those. That is a very good point. There is quite a bit of generative work, both from Gustavo Deco's group and collaborators and others that have grown out of that area, as well as Randy McIntosh and others, Olaf Sporns, that are collaborating on really answering these basic questions from that point of view. That is absolutely an absolutely important area to look at. You're right.
Yeah. It looks like your data may be able to constrain those models or falsify some version of it, or do you need to add noise? Do you need to do something else to get something closer to what you actually see in the data?
That's absolutely, yeah. That is actually a great point. Thanks, Toby, for that thought. I think that would be a great basis for collaboration with the kind of labs that do those kind of modeling approaches. And then address more specifically what quality and what quantity of noise would have to be added to account for the dissimilarities versus actually modeling fast and slow fibers, for example, as Rita is suggesting, and seeing if that better accounts for some of those dissimilarities. Of course, there has to be noise. The sources of noise that contributed to the two modalities are extremely different. Head motion and physiological noise in the context of fMRI, as well as some more recent work by Molly Bright's lab that actually shows that there might be co-vasculature responses across regions of these intrinsic networks independent of the co-activation. So it means that if I'm anticipating to start moving, and whenever I move, I know that the regions of the motor network are going to be co-activated. So if I'm anticipating that movement, maybe the vasculature is already anticipating co-innervating these areas, which will show up beautifully in the Bolt signal, but it's not of neural nature. And I don't think of it as artifact, I just think of it as a different mechanism of the brain that is not driven in the EEG. So those sources of quote-unquote noise are very different. The physiological part that's contributing to fMRI, and then of course, beyond the source leakage in the EEG, we have all the typical EEG artifacts, which are the muscle artifacts, the eye movements, and then if you have concurrent recordings of quote the massive impact of the MRI scanner on the EEG, that is also contributing to that. That said, a lot of the other labs that are doing offline MEG comparisons to static fMRI show those discordances as well. So absolutely, great idea, Dobie.
Dobby, is there time for one more?
I think so. Go for it.
Okay. Hi, Sepida. This is Bilal, part of the organizing committee for the seminar. Thank you for accepting our invitation.
Bilal, I think you got muted.
Yeah, we're waiting for your. Sorry.
Oh, sorry.
Just at the beginning of the discussion, you got muted.
Okay. So I really like the approach of using multiple methods to try and figure out what is common, what is independent. And I'm really intrigued by this repeatable observation where you had this ceiling for the correlation that you could measure around 0.3 or something. So I was wondering if, what's the status in the field about thinking about testing that with some kind of positive control or a manipulation where you evoke something like, drowsiness or anesthesia, or use a sedative or something that we know pushes the connectivity in one dimension, and then being able to resolve that with the different measures.
That's another great idea. So in addition to Dobie's model-based approach, so one would add an active causal manipulation and look how it affects the two modalities. I mean, one natural manipulation that has been studied quite a bit with concurrent EEG fMRI is sleep stages.
Yeah.
Typically, EEG is thought of as the gold standard for defining what sleep is. In terms of the connectome changes, I'm trying to see, to recall if those have been directly compared. Because again, EEG is thought of as this like state-defining kind of gold standard, but not so much in terms of affording a source-localized full-plane connectome and looking how those two things change. That's a good point. One would have to go into the literature and look at that. And of course, pharmacological interventions. I mean, the data exists. There is concurrent data with sedatives and so on that one could use. And of course, animal research, as for example, Sherlock Holtz is doing a lot of that kind of work that affords a lot more, active intervention. We are starting. We're working on recording data here locally at our hospital intracranially, and we don't have concurrent fMRI, but we have the ability potentially, depending on the patient, to do active stimulation of the cortex and see how connectivity changes. The question is if I could do that in the concurrent intracranial fMRI with our collaborators in London. I think those kind of causal interventions, yes, are the way to go. Stimulation might be hard, but in terms of existing data, yes, sleep stages definitely. My gut feeling is that it'll tell us that there will be some good level of concordance and a lot of differences again, but that's a great idea. Yes.
Thank you.
All right, I think we're out of time. So thank you again, Seppi. That was really wonderful. Just as a reminder, you have a meeting with the graduate students in 10 minutes and then with a bunch of faculty in the afternoon. We're looking forward to that. Thank you again.
Dobie, is the idea that we're eating during that meeting, or?
Yes.
Yes. Okay. All right. I'm gonna.
You definitely should eat at some point. So. Yeah, if we had you here, we'd actually give you a sandwich and you'd be eating it with other people that got sandwiches. But yeah, now you have to provide your own sandwich, but you can eat it during the meeting.
We'll do that. Thank you so much again.
Absolutely. Thank you. Bye-bye.